Hi, Michelle Glass here. Welcome back to another episode in our Chapter 8, Lecture 8 series. Our topic here is the cell cycle of the eukaryotic cell. You should consider, just like you are born and you mature and you have a job and then you might reproduce or you might not, and then you get old and you die. A cell is doing all of those same things because remember, the cell is the smallest unit of life. So whatever your organism does on the big cell scale, the cell is doing on the small scale. So that's our topic here. Now, most of the time, the cell is going to be in a phase called interphase. So 90% of the cell cycle is interphase for the cell. So this is when the cell is doing its job. This is when the cell is preparing for division, but it's not until the cell enters mitosis when the cell actually divides. And so that is the smallest portion of the life cycle. That's about 10%. And some of our cells never do mitosis. So some of our cells go into interphase and never come out. Okay, so interphase can be divided up into different stages. The first is G0. Now this is the time when the cell is doing its job. So if it is a plant cell, a mesophyll cell of the leaf, and its job is to do photosynthesis, then that cell is in G0 phase. If you're a red blood cell and it's your job to transport oxygen, then that is what you're doing. Red blood cells are an example of cells that once they're formed and they mature and they start doing their job of transporting oxygen, that's all she wrote. They just do their job and get old and die. Neurons are the same. So we expect neurons to do their job and then, ultimately they can die. Right? But they don't do cell division. But then you have cells like our skin cells that are constantly able to do mitosis. So it does vary cell type to cell type. Okay, if that cell gets a signal that it needs to divide, maybe the body is getting bigger and so you have growth factor being released to signify you need more mesothelial cells if you are increasing the size of the leaf, or if the body's getting fatter or taller if you're growing. If you have had damage to your tissue, then you might need to signal for mitosis. Mitosis is cell division for growth and repair. So any kind of signals that would tell the body that we need to make more cells because of growth or repair. When that happens, the cell gets kicked out of G0 and goes into G1. Now the G1 here is the first part of the preparation for division. So remember, this cell ultimately has to split into two. So that means it needs twice as many organelles, it needs twice as much cytoplasm, and it's going to need twice as much DNA. In G1, we're starting the making of the proteins, making organelles, and so forth to get that cell ready for division. It's going to actually stop at a checkpoint. So we have 3 different little checkpoints happening here in the cell. These are little ways to ensure that the cell is ready to move forward in the process. So basically it has to meet up with the checklist, and if it's done all the things it's supposed to do, it can move on to the next level. All of your checkpoints start out as stops, and only after the cell has, can show that it has met all the criteria will it move forward. Once the cell gets the green light, it's going to enter what's called S phase. Now this is an important part of interphase. This is where DNA replication is going to occur. Remember, as part of the preparation for division, not only do we need double the proteins, double the organelles, double the cytoplasm, but we also need double the DNA. DNA replication, remember, is how we're going to go from one chromatid to 2 sister chromatids. Remember that the sister chromatids are identical to each other and they're connected at that centromere. So even though we are doubling our total amount of DNA, we still say we have the same number of chromosomes because we are using our centromere as our count. Once you've completed DNA replication, you're going to go into G2 phase. G2 is where you finish your preparations for division. So you, again, double check, okay, DNA has doubled. Now let's make sure all the proteins and organelles in cytoplasm that we need has doubled, and we're going to get to another checkpoint. This is at the end of G2. This is called the G2 checkpoint. So again, all of these automatically start out as stop signs. Essentially, once the cell can show that it's followed all those steps, then we will see it getting the go-ahead and entering into mitosis. Now, mitosis is when we actually do our cell division. So this is that small little slice of the cell cycle. This is when we actually divide the nucleus. We're going to have an entire video just on this process, so here's just a brief introduction. Mitosis can be divided into 4 phases. So we have what's called prophase and metaphase to begin. Now, at the end of metaphase, we have another checkpoint. This is the M checkpoint. It's at this point that the cell has to, again, show that every step has been followed, and then it gets the green light, and it finishes up anaphase and telophase. There's an easy sort of memorization for the phases of mitosis. You can always think PMAT, prophase, metaphase, anaphase, telophase, and then we will go through all of this in detail in the next video. Okay, couple other things. As we talk about those checkpoints, these are basically controls on the cell cycle. So this is making sure that the cell is ready for division, that the body indeed wants that cell to divide, and that it's dividing in a very controlled fashion. Remember, our checkpoints are those stopping points. So you automatically get a stop, and then you have to have different factors and so forth to indicate that you are ready to go to the next level. There are 3 checkpoints discussed in our text. We have G1, which is probably the most important because that's going to initiate that whole preparation for division. G2 is complete when you have maturation promoting factor. And then the M phase, remember, that was required in order to transition from metaphase to anaphase, occurs when all of those chromosomes are successfully attached to spindle fibers. So as we start looking at the process of mitosis, we can see where that step is going to be critical to complete mitosis. Now, these are signals that are happening internally to the cell, but there are external signals that are important for cell division as well. There are growth factors that are released by other cells that are going to stimulate the cell to divide. So this might be something that signals for a cell to go from G0 to G1. This might be something that helps get you past that G1 checkpoint. Here's an example of a growth factor, platelet-derived growth factor. This is released by our platelet cells. So these are going to be important cells that are involved in blood clotting. So they're going to show up and release this factor when we've had damage to our tissue. So here's an example of how we can get cells to go into cell division as a result of necessary repair. Cells also demonstrate what's called density-dependent inhibition. So they're not going to grow on top of each other. So when that area is full, cell connected to cell connected to cell, then neighboring cells send a signal and the cells will stop growing. If you scoop out some of those cells, then you'll see them go back into division until they filled in that area. So this is going to be an important signal to keep cells from growing on top of other cells. And then third, you have what's called anchorage dependence. Then what we see here is that cells need a substrate to grow on. They shouldn't just spontaneously start dividing into our body fluids. Now, these 3 factors are important for us to look at and understand because this helps us to understand cancer cells. Cancer cells don't follow these rules, or they make their own growth factors, so they give themselves the go-ahead, even though maybe they shouldn't. We'll be talking about cancer cells towards the end of our Chapter 8, Part 1 discussions, so stay tuned.